Okay, thanks Isabel, and thanks the organizers for giving us the opportunity to present in this forum some of our latest results on models of motor neuron disease. As you may know, among the motor neuron diseases, amyotrophic lateral sclerosis is the most prevalent one. It's by incidence the third neurodegenerative disease after Alzheimer and Parkinson's disease. However, probably the prevalence is not so high because of the very poor prognosis and fatal course of the disease that in most cases result after paralysis, progressive paralysis of all the muscles in the body to death within a few years. Okay? This is due because, as you may imagine, there are no effective treatments available for those patients. Only palliative care is useful to prolong a little bit life and improve quality of life for some time. In this disease, there's a variety of neurons involved, but most selectively are the lower motor neurons. These are the big neurons that are located in the spinal cord and brainstem, and that innervate our skeletal muscles, allowing us to make any movement. But also there is involvement in the upper motor neurons located in the cortex, in the motor cortex. Okay. The vast majority of the cases are of unknown etiology. So they are termed like sporadic ALS, whereas a percentage that classically has been considered between 5–10% of these cases have familial type of evolution because of mutations in a variety of genes that are also being discovered, moreover. This percentage certainly is under re-evaluation since 2011, when there was discovered a higher prevalence of about 20% in some studies, some cohorts, of abnormalities and exonucleotide repeat in the chromosome gene C9 or 72. Okay, however, for most of our knowledge until now has been based on the development of some transgenic mice based on mutations that have been previously discovered in some human patients. And the most relevant and most significant type of this transgenic mice is the one with the human mutation in the superoxide dismutase 1 gene, SOD1 gene, that makes a variety of mice depending on the location of the mutation. When the location is placed in the locus 93, these SOD1 gene 93 mice are really probably the most used in the literature in previous studies to reproduce the model in humans. As you can see in the cartoon, these mice quite well reproduce the pathology in humans. They are quite well functioning until about 3 months of age, when they start to show signs of dysfunction, locomotor falls, and then paresia, and then come to paralysis, and then between 4 and 5 months, they die. Okay? So based on these models, many people have been able to investigate many of the etiologic mechanisms involved or linked to the ALS disease. And there is a whole variety of things. As you can see in this schematic summary, there are involvement of oxidative stress problems, mitochondrial dysfunction. There is also excitotoxicity playing a role, abnormalities in RNA processing. So endoplasmic reticulum stress is involved, and the aggregation of toxic proteins, among them SOD1 and TDP43 being the most relevant. All these potential mechanisms play a role in deriving in the death of the motor neurons and the subsequent clinical symptoms. Using also these models, these animal models, more than 100 of studies, preclinical studies, have been performed, trying to target some of those etiologic mechanisms for therapeutical approaches. Okay? Between all of them classified as relevance in the success of the results, about half of the studies revealed some positive effects in terms of animal performance, and most importantly, animal survival. That has been considered a key mark of the potential evolution. However, no one of those studies in the literature has achieved to make those animals to survive longer than 25%, the classical. And this 25% has been, by some consensus, established like a target. If you succeed to overpass this limit, you have a big success, and you may apply to get the prize for life, about $1 million. Okay. Then why this is happening, why so many failures? Certainly there are several reasons that can explain. But certainly for a disease with so many etiologic mechanisms, with so many cellular and molecular pathways involved, it seems quite difficult that a single targeting, a single mechanism will result in completely improvement of the case. So with the variety of molecular pathways and the variety of cells, motor neurons, not only motor neurons, but also skeletal muscle, inflammatory cells, glial cells, also showing abnormalities in ALS. Certainly one hypothesis that has been pushed forward and we have followed it is that therapeutical combinations, trying to target several of those mechanisms involved, may have better chance of success, since we don't have a cure because we don't know the real etiology of the disease. Then in previous forums of CIBERNED, we had the chance to present some of our previous results. The first one was very promising. We addressed the activation of the sigma-1 receptor. This is a very kind of peculiar receptor that is highly expressed in motor neurons in the spinal cord and is linked to several neuroprotective mechanisms, such as reducing calcium influx in the cell by different pathways, also reducing the activation of oxide nitric synthase, and also modulating the microglial inflammatory response. So with a single drug, we could address several mechanisms. The results, to make the thing short in one slide, were very promising. We showed by electrophysiological studies that the animals treated with the agonist that was named pre-084 maintained quite stable the neuromuscular function in contrast with the non-treated mice. Also, the animals were able to survive quite longer, about 10–12% longer than in controls. And that was correlated with a higher survival of the spinal motor neurons by about more than 50% extra of the numbers. Okay? However, there was still insufficient. So we moved to another multi-target type of drug that could be useful, and that we presented the results in the forum 2 years ago. That was resveratrol. Resveratrol is a neuroprotective drug that has been used in other type of pathologies and models, and that acts through different mechanisms, mainly by pathways mediated by sirtuin-1 and AMK. Okay, so they include several potential neuroprotective actions. Our results were similarly promising. As you can see here, the neurophysiological studies also showed maintenance of the compound muscle action potentials, indicating maintenance of neuromuscular function. The overall locomotion was also maintained. Okay, survival was increased again by about 10–15% of the lifespan of the mice, and the proportion of motor neurons surviving in the lumbar spinal cord was significantly increased. Moreover, we showed evidence that mitochondrial function that is quite abnormal in the SOD1 mice was restored to a normal type of oxidative processing in the mice. So there was good expectations. So at that time, we had two potential mechanisms targeting different pathways to promote motor neuron survival. As someone asked in the audience at that time, whether you don't mix those two therapies. And certainly we were doing that, but now we have the results. So we treat the animals with resveratrol that's given incorporated in the diet, so continuous intake, more than you can drink in wine, so you need to get a special amount, and also daily injections of the pre-084, that's a sigma-1 receptor agonist. Individual groups, individual treatments, and combined. The animals were treated from 8 weeks of age when we can diagnose these mice by electrophysiology and follow it over time with final harvesting of samples at 16 weeks, well advanced at the stage, and some cohort follow it for survival. Okay. Well, the good part was that we were able to reproduce our previous results with the sigma agonist and with resveratrol. So there was consistent results. And as you can see, in blue lines for the pre-84 and in red lines for the resveratrol, the rotor-rod performance of locomotion was improved. The neuromuscular function indicated by the compound muscle action potential was again improved, and survival was longer. The bad part is that adding the two treatments did not provide any synergistic additive effect. So as you can see, the red lines that are the combined treatments produced similar improvement than the single treatments. Why did it happen? Well, we don't know. You can hypothesize. We can discuss many potential reasons. One is that the model itself has some limitations, that we will never be able to make the animals live longer and survive longer, well, for other reasons that we are not really knowing yet. But other option is that possibly we are overlapping part of the pathways, that adding more in the same basket will not really become much better. So going back to our schema, we have been able to really target several of the mechanisms that I pointed before. Improved mitochondrial function with resveratrol, reduced excitotoxicity, improved endoplasmic reticulum stress, and despite all these cumulative actions, neuronal survival is not more improved. So we thought about going to other target cells. And one of the logical cells, especially after hearing the previous talk, was to go to neuroinflammatory processes. Microglial cells' reactivity and role is a hallmark in all the neurodegenerative diseases, and also in ALS. So the increased amount of infiltration of microglia and also of lymphocytes is present in the spinal cords and also in the cortex of those cases, in animal and in human cases. So given the rural role of microglia that has been pointed out before, so I will not stress longer, between two phenotypes, a possibly protective M2 phenotype that could be acting at the beginning of the course of the disease, and a more toxic M1 microglia phenotype that is completely predominating at the later stages of the disease and contributing to neuronal degeneration. So in cooperation with the group of Gomez Nicola and Hugh Perry in Southampton, that they have shown that previously that by blocking the colony-stimulating 5-HT1 receptor activity with a specific drug, GW2580, was good effects in other neurodegenerative models caused by prion disease and by other groups in multiple sclerosis. We tried to use this drug. This could specifically block the CSF1 receptor and thus modulate microglial proliferation and potentially promote the expression of the M2 protective microglial phenotype. So first of all, we wanted to check if the CSF1 receptor was relevant for the disease in the model. So the presence of CSF1 receptor was checked in the spinal cord. It was present and mostly was present in microglial cells. And also the levels of mRNA for the receptor were increased in the SOD1 mice, not significantly at 12 weeks of age, at the beginning of symptomatic stage of the disease, but quite more at late advanced stages of the disease, 16 weeks. Also the ligand of the receptor, CSF1, was increased at RNA and protein levels in the spinal cord of these later stage mice. Okay? So by using the same schema, we treated the mice daily with this GW drug from 8 weeks until 16 weeks and periodically tested the animal with electrophysiology, locomotion tests, and finally some molecular and immunohistochemical tests. The results showed again that we were able to protect plantar muscles in the paw, tibialis anterior muscle, potentials in the leg. They were stable from about 10 weeks of age. After 2 weeks after treatment, they remained with very low declination, with respect significantly better than in the control non-treated mice. Rotarod was also improved, but not so much as the electrophysiology. And the forced locomotion also showed some good signs at the later stage, at the end of the treatment. When looking at the spinal cord, so the number of motorneurons, these large purple cells, was increased in the treated mice. You can see about more than 10% increase. And survival was also increased, especially at the final stage of the disease, curiously. So the GW, by modulating microglial cells, was able to improve the outcome of the animals. And that was correlated with an effect on microglial cells in the spinal cord. And you can see by flow cytometry and by immunohistochemistry, we could see that by 16 weeks of age, the presence of total microglia was significantly reduced, okay, and also by immunohistochemistry, but not at 12 weeks, curiously, and the treatment started at 8 weeks. So the effect of microglia was relatively late, later than the beneficial effects that we already detected by 10 weeks of age. So there was some discrepancy between the morphological effect on the inflammatory cells and the functional effects that we saw in the full animal. That's why Ruben Lopez-Valles, who is here, pointed that probably there was another effect laying down here. And so he checked in the lab the presence of macrophages in the periphery. From samples in the peripheral nerve, those are also increased in the infiltration of macrophages because motor axons are degenerating also in the peripheral nerve. So the presence of macrophages is highly increased in these animals at 12 weeks, at 16 weeks. And there, certainly, the treatment with GW reduced the number of macrophages in these samples already at 12 weeks, and also more at 16 weeks. And the effect of the drug was markedly proved in the periphery, because by flow cytometry, they could show that monocytes in the blood of these mice also showed a clear reduction. So the drug was, with a novel result, acting not only at the central level on the neuroinflammation, but also the peripheral level, and it's probably the first report that shows that a positive effect of modulating the neuroinflammatory response in the peripheral nervous system in this model of animals. Okay, so we had something to add to our other previous targets pointing to the motor neuron survival, but still, probably it's not going to be enough. We don't have mixed yet all these 3 treatments. That will be probably take longer. But we wanted also to tackle other cells, and going more to the periphery, as we have shown that at the level of the axon, there is important actions to take. We went to the end, the neuromuscular junction. The neuromuscular junction, the muscles and axons interact and form a synapse, but that were also the terminal Schwann cells, a peculiar type of glial cell, is also contributed. So we wanted to address this point, because it has been already described in ALS patients and in the SOD1 mice that neuromuscular junction become denervated. The axon withdraws from the synapse, as you can see here, partially and completely denervation of the end plates, quite before the motor neuron dies. So it mimics a little bit what the neurologists working in the PNS know, like a dying back phenomena. So the terminals is withdrawn, and at the end, the soma is going to die. So one of our points was maybe an early intervention to present the attachment, the neuromuscular synapse with the muscle, may protect back the neuronal soma in the spinal cord. To that end, we decided to target a trophic factor that is neuroregulin. Neuroregulin is a full family of trophic factors, but one of the main effects has been always known to support axonal neuromuscular junction formation during development in the early stages of the life, but also in the maintenance of these synapses. There have been also some previous papers describing contributions of the neuroregulin addition to improve axonal regeneration, and also some effects on the Schwann cells for myelination and linked to the axon. So a couple of reports also supported the possible role of neuroregulins in ALS pathology. Those were described in 2 papers, previous papers, one showing reduced levels of neuroregulin 1 type 3 expression in the spinal cord in patients, and also in the SOD1 model. And another that's a dysfunction of one of the receptors, RBB4, where neuroregulin 1 can link and make the effects in human ALS patients. So first of all, we wanted to see if alteration of this neuroregulin RBP4 pathway was existent in patients and in our animal model. By checking in the mice, you can see that the RNA expression of RBP4, this special receptor, in gastrocnemius muscles was significantly decreased from normals in the mice already at early ages, 8 weeks of age, when no symptoms apparently appear in the animal. And also there were decreases in the soleus muscle. The soleus is more protected muscle. The gastrocnemial muscle, the type of motor neurons, has a faster decline of function. But in the soleus muscle, the levels were also decreased, but a little bit later, so that fitted with the disease. And the RBB receptor could be located most likely at the level of the terminal Schwann cells. It's very difficult here to detect by some immunohistochemistry this type of receptors. RBB2, another important receptor in the muscle, was not affected in the mice. So we obtained some muscle samples from patients and also showed that all the cases had significantly reduced levels of RBB4 expression at the mRNA and also the protein levels. So the abnormality appeared to be quite relevant in patients and in animals. And then we pointed out how could we induce an increased activation of the RBB4 receptor to compensate for this effect. To that end, we make a viral vector, an AAV vector, to overexpress neuroregulin 1 type 1, and injected that in a muscle. We selected the gastrocnemius muscle in the back of your leg. Okay? And after a couple of weeks, you can see that the expression of neuroregulin was dramatically increased in these muscles, as you can expect. And then assessed the function. From an electrophysiological point of view, the gastrocnemius muscle maintained the amplitude of the potential, all the function, over time after a couple of weeks of the transduction needed to really transduct the tissue. Whereas the tibialis anterior muscle, that from those who don't know anatomy, is in the front of the leg, so it's just very close, okay? That was not injected, didn't show any change. So we were able, with this viral, to really make some difference in the injected muscle. And that was due to an increase in the size of the individual motor units. We didn't make new motor units, new motor neurons arriving, but we were able to increase the size, the number of muscle fibers innervated by each axon in the injected muscle. Okay? So that was an important point. And then by some histology, we could see that certainly we had not affected the number of motor neurons. Motor neurons died the same, and the number of axons in the spinal roots were also the same. But when looking at the muscle, the number of innervated end plates, and you can see in green the end plate, part of the muscle in the synapse, and in red the axons. So the number of end plates was increased significantly in the injected muscles with respect to the controls. So we were able to see also by immunohistochemistry that in the treated animals, those axons that appeared in the muscle were able to make new branches, and by those new branches re-innervated new muscle end plates. So each motor neurons were able to innervate more muscle fibers. So the strength could be recovered, okay, by the same number of motor neurons remaining, and that also quantified significantly. Then to prove that we were acting and where we were acting and how is it going, we checked some of the molecular pathways that could be linked downstream of RBB4 targeting. RBB4 phosphorylation was increased in the muscle of the injected animals, so that and phosphorylation was indicating that. And also these Schwann cells had a higher expression of ERK1/2 activation. Whereas in the muscle fibers, we found selectively an increase of AKT activation. Okay? So that proved that the neuroregulin treatment acting on RBB4 was able to activate the terminal Schwann cell, that is a key cell to promote synaptogenesis in the muscle, to activate these pathways, and also was able to activate the muscle fibers to probably help also the motor axons. So this is certainly at the starting, hopefully, point to make something that is a new approach. Instead of trying to protect the neurons don't die, we are trying now to improve function in the affected individual by increasing the neuromuscular function at the periphery, and certainly by combining the periphery and some central approach could be a new hope for those patients. So, it may be difficult to inject with this vector the more than 300 muscles that all of us have in the body. Okay, so that was our proof of concept that by injecting a muscle, this muscle was really improving in function and in perspective of reinnervation. So now we are trying to develop a viral vector that could be applied endovenously and by the linked to the desmin promoter, go to all the muscles in the body and being able by a single injection to tackle all the perspectives. And we hope to be able to show you something in a next forum or next occasion. Thank you very much.